Good afternoon, everyone. It's my privilege to be here where I'm an alumni of this wonderful university and this alumni of this wonderful college of this main campus. We used to say proudly main campus in our time. So, and moreover, I'm a local because my house is in PN Pudur only. So, this is a very good opportunity for me to be present this topic in this Augustus gathering. Uh, if I supposed to brief about myself, already Madam has given a brief introduction. I was born in Theni district of Tamil Nadu. That means Podi Minachuram, Theni district. I have completed my undergraduate schooling from Theni district. My undergraduate is from the very prestigious College of Agriculture Research, Agricultural College and Research Institute, Coimbatore. And after completion, I have completed my master's and doctoral at uh, HP, Himachal Pradesh Agriculture University, Palampur. You all must be knowing the Solan. In Himachal Pradesh, there is Agriculture University. Also there, it is in Palampur. So I have completed my degree and master's and doctoral from there. In my master and doctoral degree, I have done in tissue culture and in maize and doctoral. I have done it double applied breeding in wheat. Following molecular cytogenetics, my specialization. Of course, now I am not doing, but my work was there. Then after that, I have joined in Central Agriculture University, Imphal, Manipur. I have worked for 12 years as a pulse breeder, and I have a nice time to work with our Madam Germany. Almost 12 years we worked together in this pulse breeding. After that, I have joined. Now I'm working as associate professor genetics and plant breeding, and it's Agriculture University, Jodhpur, Rajasthan. For the past 2 years, I'm working there. So I can say I may roamed almost around the India. So now this molecular cytogenetics and double haploid breeding for crop development, mainly this molecular cytogenetics in India, it is the work is very less. I can say, not more people are doing for the past. Even when I was published my work, even after 10 years, that is a fresh work. There is not much from India. Not much people are working in molecular cytogenetics. Very few actually working. But in USA or Germany, or if you take it in European countries, the people are working molecular cytogenetics also one of the field combining with the molecular genetics. So in this background, just I am going to just brief about the what are the methodologies are in molecular cytogenetics. Double applied breeding can do it in a different way. If you can go anther culture, you can embryo culture, and you can go other microspore culture, whatever things we can go. But the thing is, I worked in wheat. So on the background of that, I'll discuss about this molecular cytogenetics and the double applied breeding. So if you know the term cytogenetics, is referred to the study of genetic consequence in the terms of chromosome number, structure, and behavior vis-a-vis species and evolution. Somebody can come and stay. Just can you operate? So if you come in, it's a general cytogenetics terminology. If you come for plant cytogenetics, the field of plant cytogenetics heavily induced by Barbara McClintock. Everybody know it is some jumping genes. It's given by Barbara. She worked in a cytogenetics. From that cytogenetics, she used acetocarmine staining. Through that, she identified all the individual chromosome from a single meiotic nucleus with a combination of two metrics. That is, when we go for the cytogenetics, normally we look after the relative length and arm ratio of the chromosomes. That's the main. Then her studies unequivocal identification of individual chromosomes established a milestone of the scientific community, which allowed new discoveries regarding the dynamic structure and behavior of maize genome. Further development of chromosome bending techniques greatly increased this study of the cytogenetics. Then Kasperson et al. Proposed a Q bending technique. We know all Q bending. And Vosa and Murchie compared the Gimsa C bending to Q bending on the chromosome bean and killed garlic and maize. So further Gimsa staining technique also showed its utility to identify individual rice pro-metaphase chromosome, karyotype development for diploid rye and barley, With the advent of information on DNA and its characteristics, modification of DNA staining dyes and binding techniques were adopted and optimized cytogenetic characterization of different plant species. The classical approach have been proven invaluable. We come to the modern technique in situ hybridization, which allow for direct visualization of the specific DNA sequencing on chromosomes. Coming to the molecular cytogenetics, a combination of classical cytogenetics and recombinant DNA technology gave birth to the versatile multicolored fluorescence engineering based chromosome biology called molecular cytogenetics. During initial development, we have used radioactivated isotopes. Those isotopes is to handle this radio isotopes are very difficult. To replace that one, non-radioactive isotopes are like non-radioactive heptanes are used. Heptanes such as biotin become available to adapt widely. Using these advantages, increasing stability, safe handling. When you using this biotin, this heptane, non-radioactive heptanes, increase stability, safe handling, rapid, precise spatial localization, less background, and most importantly, the ability to use multiple colors on the single chromes preparation. Then what is the in situ hybridization? The development of in situ hybridization techniques opened up opportunities for cytogenetic analysis of any species, regardless of its inherent chromosome morphology. In plants, the use of radioactive tagged or modified nucleotides labeled with biotin, digoxinine, or fluorescent heptanes, and fish probes also permit microscopy visualization and localization of complementary sequence in cells, nuclei, and on individual chromosomes. A difference between this marker-based and this molecular cytogenetics, visually you can see where your particular chromosome segment is located. That is one beautiful thing in this molecular cytogenetics. Then fluorescent, what is fluorescent in situ hybridization? This is a molecular cytogenetic technique, allows cytogenetics to analyze chromosome resolution at the DNA or gene level. This can be performed on dividing and non-dividing interface cells to identify numerical and structural resulting from genetic disorders. This piece compared to plant, mostly in medical field, like cancer, all those things, chromosomal aberrations, abnormalities, this fish and GC is mostly used. Coming to the basic piece, makes use of green and red fluorochromes for probe detection and DEPI, that is diamino 2-phenylindole, for counterstaining the chromosomal DNA. Although FISH is commonly used to map unique or low copy number sequence, however, it also showed its potential to localize repetitive DNA sequence. Probes are tagged with the fluorescent nucleotides either directly or indirect. So this is done by attaching nucleotide to small molecules such as biotin, digoxinine, and dinitrophenyl, to which fluorescent antibodies can later be bound. The cells are then viewed with a fluorescent microscope. The fluorescent signals represent the probe that is bound to the chromosome. When you are just attaching this probes attached with the fluorochromes, that will attach to a particular sequence. In that sequence, actually, when you are visualizing through the fluorescent microscope, special microscopes are there, fluorescent microscopes. Through that fluorescent will become, so you can observe where your probes are located. So use of FISH in crop improvement, FISH has been found most successful in mapping repetitive and single copy DNA sequence on pro-metaphase chromosomes, interface nuclei, packaging complements, and chromatin fibers and naked DNA molecules. Then here, accurate location of repetitive and random arrays plays a major role in chromosome identification. The broad application of FISH is structural, comparative, and functional genomic place plant cytogenetics in an important place to complement. Genomic, here another is genomic in situ hybridization, where fluorescent microscope we observe the particular chromosome segment we can observe. Here in genomic in situ hybridization, you can observe entire genome. Like wheat, if you take it, we will take with the entire Y chromosome with the green chrome. That will be exactly go and attach with all this painted with the Y chromosome. The entire genome of Y chromosome we can observe. So later I will show the slides of the slides you can observe. So advances molecular cytogenetic techniques, this is an indispensable tool in nowadays in the genomic research. It's an indispensable tool, and rapid development in genetics, molecular genetics, molecular biology, and genomics, together with molecular cytogenetics, have driven major conceptual advances. A basic development was the use of several colors for labeling. You can use it several colors to paint the entire chromosome. Though there is a method, dry fish was developed to improve the detection sensitivity of fish experiments. The method allows signal amplification by using a peroxidase conjugated antibody. The fluorochrome labeled tyramides will be go and attach. The specific sensitivity will be signal recognition sensitivity will be improved. Then DNA fiber fish, make it a DNA as a fiber with even single copy of genome also you can observe through the methodology. Use of fiber fish, this plant, in this actually exact position and ordering of the DNA clones are needed. It can be evaluated distance and overlaps between neighboring sequence. The minimum target size that can be distinguished ambiguously in this in plants is 10 kb. You can target it. And three-dimensional 3D fish, you need to take a photograph, taking the photograph one by one of different chromosome like that. Then you can observe the 3D. Then fish on superstretched chromosomes. In interface nuclei, superstretched mitotic metaphase chromosome and meiotic packaging chromosome provide intermediate resolution power for fish mapping. The relative position of cloned separated by more than 100 kb can be resolved on these cytological targets. Packaging chromosomes are particularly versatile target for fish mapping. On the other hand, flow-sorted plant chromosome at meiotic metaphase can be stretched to more than 100 times of their original size. The fish on stretched chromosomes showed brighter signals on the untreated control, presumably. Fish on superstretched metaphase chromosomes provide a mapping resolution up to 70 kb. This modification of fish provides an alternative mapping target for those plant species where meiotic packaging chromosomes are not suitable for cytological studies. And it means bacterial artificial chromosome fish. This genome-wide sequencing project, a genomic library holding large DNA fragments, is an important tool for physical mapping or positional cloning of important chromosome landmarks. Backfish, a unique tool for molecular cytogenetic, uses genomic DNA cloned in large insect vectors such as bacterial artificial chromosomes, a win combination of fish. The back clones provide efficient resources for chromosome-specific fish markers, especially for plant species having small genomes such as rice, cotton, and sorghum. Backfish favors a large clone as a probe for better relation. Molecular cytogenetics in plant genome research. In physical mapping and in situ PCR, plant genomes are known for abundance of repeat sequence, and cytogenetic or physical mapping of such repetitive DNA sequence decipher their genomic distribution and precisely identify the typical chromosome or set of chromosomes. These repeated rDNA genome clusters are being widely used under common starting point for fish-based mapping. The two types of ribosomal RNA genes, that is 18S, 5.S, and 26X rDNA and 5S rDNA, have been extensively used as probes for physical mapping in higher plants due to their arrangement in tandem arrays. Now here you use it in hexaploid with 6 loci of 5S rRNA genes were identified on the short arm of the chromosome of homologous group 1 and 5, that's 1A, 1B, 1D, 5A, 5B, and 5D, whereas 18S, 5.S, and 26 rDNA loci were mapped on the short arm of 1A and 1B and 6B and 5D chromosomes and the long arm of 7D chromosomes. The rRNA genes are associated with the nuclear organizer region, and the visualization of such repeated sequence clusters at interface represents the number of active rDNA loci. You see this multicolor fish in a diagram. If you see it, mapping of 5S rDNA and 18S, 5.8S, 26 rDNA genes on the chromosomes are bred with to the fluorescent signals can be seen for 5S. Red color, if you see red color, if you see red color, that is 5S nuclear organizing region. Multicolor fish. In this fish, multicolor fish approach targeting repetitive DNA and rDNA probes also serve as chromosome identification markers in many plant species, for example, common wheat. Here in this figure, 7 color fish on metaphase cells on common wheat, 7 DNA sequence, that's probe sequel serial 119.2, probe sequel serial 74, probe Agiolophis sporosa AS1, telomere, and 18S26 rDNA, 5S rDNA, and gliadin were detected by red, bluish green, green, orange, pink, and white fluorescence. All these multicolor fish. Here in this chromosome, the basic principle of fish was further exploited to paint individual plant chromosomes. You can individual plant chromosomes, you can paint it in different color. That's a means chromosome painting with the different probes. Use of chromosome painting. Chromosome painting provides insight into genome duplication, multiplication, and karyotype evolution closely related taxa. In mostly we use for Arabidopsis fish, chromosome and segment specific probes were hybridized to paint the chromosomes from species related to Arabidopsis thaliana and Brassica species. These experiments proved that the technique is feasible for the detailed investigation of pairing behavior of homologous chromosome during early prophase, because this chromosome pairing and other thing, which chromosome is pairing with which one are crossing over, with this painting we can easily identify one. And alien chromatin and transient detection. In the alien interchromatin, the same thing. We will when we go for the cross of a wheat and triticale, we can identify the rye chromatin in the wheat background. It means different probes we can use it like means rye probes are there, and with that the probes we can identify. Modern molecular cytogenetics, the biological question may be not be solved by simple localization of DNA. When we go for that with the protein, we need to immuno fish, we can use it. Several plant laboratories have developed techniques that combine fish with immunoassay methods. Such modernization of cytogenetic technique involves an immunoassay of specific antibodies and a cytogenetical preparation followed by standard fish procedure. Immunofish has been used to reveal DNA methylation and histone modification with a specific genomic reason. This figure shows that one. If you the immuno staining of chromosomes of alien species using anti-Fc H3 antibody, DEPI staining is blue color is a DEPI staining chromosome blue, and the visualization of immuno signals of anti-Fc H3 antibody in red color. Coming to the double haploid and means double haploid, how we can use it double haploid, and the double haploid following by double haploid molecular cytogenetics, how we can use. My specialization in my PhD, I have worked in the double haploid breeding in the wheat using chromosome elimination technique. So I will discuss further, maybe another 15 minutes. So coming to the means in wheat, you must know in wheat means this chromosome elimination technique is first means worked in the barley, Ardium bulbosum and Ardium vulgare. The selective elimination of the vulgare genotype means chromosomes. Then we will means haploid. Then we will double it. That's first initially worked out. Followed one in 1987 and 88. Laurie and Bennett, they worked in chromosome elimination technique using maize pollen in wheat. So when we are putting the maize pollen in the wheat, that will be the fertilization will takes place, and the first and second mitotic division automatically the chromosomes of maize will not pair with the wheat. Automatically it will selected, it will be eliminated. Then when it go to second, third mitotic division, only wheat chromosome will be there, only haploid nature. So that will be using artificial. When the haploid plants will be there, you need to give the colchicine treatment in the root in ground root initiation stage. Then further which is developing, whatever trillers are coming, that will be doubled. In that way I have worked in that. Instead of using maize, we have used the one grass. The grass, it is available in the cold area, Gogan grass. That is scientific name is Imberata cylindrica. Using this Imberata cylindrica, when you are using this pollen of Imberata cylindrica, there is advantage over the maize pollen, because when in the cold season you can't grow the maize. You need to grow the maize in the polyhouse, because maize need the warm temperature. So, but this Imberata cylindrica is readily available in those area when the wheat is coming for the flowering. So that time we can collect this pollen, because cost efficient, because naturally available abundantly. So we will take this pollen and we emasculate the wheat in the first day, means first day. Next day early morning we will go and collect this pollen, and we will pollinate. Instead of maize pollen, we will use the Imperator cellerica pollen. So after that, next day, after 24 hours of the pollination, we need to give the injection of the 2,4-D. 100 ppm of 2,4-D in the case of wheat into wheat cross, 250 ppm of 2,4-D solution in the case of when you are involving the wheat into critical cross. So after 14 days, if you observe the pseudo serial. I can so. This is the entire process of the double diploid production for these using Imperator cylindrica and both maize and Imperator cylindrica. So when we are emasculating pollinated, next day you need to go and means the first winter nodal, you need to give the 2,4-D injection. Like means anything to improve, we are giving injection. Same way in plant also you need to give an injection to induce the haploid. So after 14 days, automatically if you means harvest the spike, after 14 days you can harvest the spike. So when you are harvesting the spike after 14 days, so you observe against the light. If you see the diagram of the 8th diagram, we are 2,4-D injection. After that your means spikes are there. After the spike can be harvested, you take to means laboratory. From that you need to take the pseudo seed, we can say. Pseudo seeds will be formed properly. Absolutely there is no problem, but only the fluid used to be there. Some, if it is properly fertilized, some the haploid embryo will be there. Those haploid embryo you need to sort it out against the light. If you see, if the embryo is there, you can see, I mean, you, very minutely you see one dot. If you see the 8th diagram, 8th means picture, if you see it means in the middle of the forefinger and the thumb, it means it's rescued that embryo, automatically it will be distorted, it will be die, because up to some particular tile that will use that solid solution is there. Solution is there, that means the solution that will survive. After 18-20 days, if you go, it will be abort. So we need to rescue, that is embryo rescue. We need to rescue the embryo, and we need to prepare a slanting medium. With that medium, you means we need to culture it. You can see the it is in the 11th diagram picture. You see, after culturing, we need to keep it in the 4 degrees for 3 days, means 48 hours. 48 hours we need to keep it in a dark in the 4 degrees C. So it will be once it started germinating of the embryo, haploid embryo, you need to change it to growth chamber. For the in the dark, it should be kept it in dark. After the dark, it will be coming to, when you shift it to light, it will be green pigment. It will be absorb the green pigment, means using light absorb the green pigment. It will start grow. These regenerating haploid plants will be kept it in the means for 25 days, the plants. Then after that you need to take to, you need to prepare as means with filter paper. You need to prepare a V shape structure, and you need to plant the remove from this medium, and you need to put it there. Through that solution, it will absorb and it will grow. When fully grown plant will be transferred to the means haploid plant cell after colchicine. After growing from this test tubes, it should be removed and it should be given the whole season treatment. It will be 0.75 ppm. You need to give with that cold season treatment for 5 hours. That's then after that you need to shift to the so means soil solution, culture means soil culture, and you need to plant it, and you need to shift to the means cage house. So once after that, really the doubling with the cold season doubling happen, the several tillers will come. Really the doubling is happen. Those tillers only, the spike in that grain will form. Everything will structure, spike will come. Once it is doubling happen, that spike will be having the seed. So in that method, we have developed almost in my PhD study, I have developed 12 double haploidy. Further, after that double haploidy, these are the some of the examples for the double haploidy which we use for the molecular cytogenetic studies. So here you can see there is mainly in wheat, if you go for wheat, mainly people looking after the 1BL 1RS translocation. When with this translocation is having the right genome that is conferring the resistance of leaf rust, all rust resistant genome, the genome and the drought tolerance, all those things, particular characters, this 1BL 1RS translocation is giving. This 1BL 1RS translocation you can see with the red color, critical with double applied bread wheat line with probe bioassay AS1 green. Green color, it shows the PAS1 means Agelopis crossa, and DIC PSC sickle cereale, that is red. Wherever red color is there, that is there is a translocation, 1BL 1RS translocation is there. When the first chromosome, B chromosome, in that one short arm of right chromosome is translocated. These are the some substitution, detection of R substitution in all green color. If you see third C diagram, if you see there is a total substitution is takes place. If you see the third entire green color with R, substitution is there. And coming to the D diagram, if you see here, one pair of total chromosome of Y is transferred. Genome, total genome. Here also same detection, 5 pair of Y chromosome is transferred in the green color. Similarly, 4 pair in the diagram A and diagram B. There is a 4 pair of Y chromosomes. This is genomic in situ hybridization. One is in the previous slides, first A and other, that is actually a fish. This other second, all this genomic in situ hybridization. Entire genome you can observe. This is all about the molecular cytogenetics and double applied breeding. Thank you very much for this opportunity which provided by this Honorable Vice Chancellor of this Tamil Nadu Agriculture University and Dean PGS, Dr. Sindhu. I thank the all August gathering. Thank you very much, and I thank my teacher, Madam, your HOD. Maybe she may not identify me because we know. Thank you very much, my dear students, for patiently listening me. Thank you. If you have any query, you can we can just discuss. Whatever I know, I can discuss. Because in India, I already told you, in very few labs are working. Still, because this is a wonderful, because it takes time, lot of time. Patience you need, slide preparation you need to do. After you need to treat the slide. All this is actually 1.5 day, 2 days. One slide, if you want to observe with the fluorescent microscope, you need to at least spend 2 days. After all preparation with the preparation and hybridization, all those, if it will go lengthy process of 2 days. After that only you can observe what are the things are there. You need to prepare a lot of slides, a lot of plans. But it's worthy because now IRI people are doing, very few general universities are doing. But in agriculture university, very few people are doing. Only very few, I can say. Double applied bidding in here rice you are using. I think Andhra culture you must be using. Chromosome elimination nobody is using, I think so. In maize also people are using inducer. One inducer line is there with that double applied breeding they are using. Nowadays we are speaking of speed breeding. So people may not be doing double applied breeding, but speed breeding people are thinking within a season they can do it. And another way is subtle breeding. Double applied breeding because double applied breeding, you know the advantage, you have all studied, I think so. Advantages and disadvantage. Advantages is within 1.5 year, 2 year, you can develop a line. The doubling homozygous line, you can get it instead of 6 year, you can get it within a 2 year. You can develop the double applied line. So in that we can go for the other further cytogenetics mapping. You can go for a mapping population, like immortal mapping population. You can generate. So likewise, this double applied breeding will be used. Combined with this molecular cytogenetics, it will be a good one, good option. Madam, I asking in this, no, madam, please. In this, here we have means fluorescent microscope. We have. Okay. Thank you. Thank you very much for this opportunity.